DNA replication. DNA replication is the making of an exact copy of the DNA molecule. The replication is made by a semi-conservative method, which was predicted by Watson and Crick and proven by Meselson and Stahl. It is regarded as semi-conservative because each of the resulting two molecules consists of one old strand and one new strand. The descriptions of DNA replication is as follows. 1. Proteins and enzymes assist in replication of DNA. Replication begins at special sites called origins of replication. The replication bubbles form. Helicase enzymes unwind the double helix and unzip the two parental strands by breaking hydrogen bonds. The separated two parental strands act as templates. Single-stranded binding proteins add as scaffolding, holding the two strands apart. Topoisomerase lessens the tension on the tight helix. 2. At each end of the replication bubble is a replication fork, a Y-shaped region where the new strands of DNA are elongating. 3. DNA polymerase III cannot initiate the synthesis. The pre-existing chain actually consists of RNA primer produced by the enzyme called primase. As two strands of DNA are anti-parallel, they are oriented in opposite directions to each other. Therefore, the formation of two new strands must also be anti-parallel to their template strands. DNA polymerase can only add complementary nucleotides, C with G and A with T, to the free 3 end of the primer, never to the 5 end. Thus, a new DNA strand can elongate only in the 5 to 3 direction. 4. DNA polymerase replicates the two original strands differently. Although it builds both new strands in the 5 to 3 direction, one strand is formed towards the replication fork in continuous and linear fashion. This is called the leading strand. The other strand, the lagging strand, forms in direction away from the replication fork in a series of segments called Okazaki fragments. Each Okazaki fragment is about 100 to 200 nucleotides long, and then they are joined into one continuous strand by the enzyme DNA ligase. 5. DNA polymerase I carry out mismatch repair, a kind of proofreading that corrects errors. Damaged regions of DNA are excised out by DNA nuclease. Nucleotide excision repair of DNA damage. 1. Teams of enzymes detect and repair damaged DNA, such as this thymine dimer often caused by ultraviolet radiation, which distorts the DNA molecule. 2. A nuclease enzyme cuts the damaged DNA strand at 2 points, and the damaged section is removed. 3. Repair synthesis by a DNA polymerase fill in the missing nucleotides. 4. DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. 6. Each time the DNA replicates, some nucleotides from the chromosomes are lost. To protect against the possible loss of genes at the end of chromosomes, eukaryotes have special nonsense nucleotide sequences, TAAGGG, at the ends of chromosomes that repeat thousands of times. These protective ends are called telomeres, which are created and controlled by the enzyme telomerase. So every time DNA replicate, the telomerase gets shorter. Enzymes that take part in DNA replication and their function. Helicase unwinds parental double helix at replication forks. Single-strand binding protein binds to and stabilizes single-stranded DNA until it is used as a template. Topoisomerase relieves overwinding strain ahead of replication forks by breaking, swiveling, and rejoining DNA strands. Primase synthesizes an RNA primer at 5, end of leading strand, and at 5, end of each Okazaki fragment of the lagging strand. DNA polymerase III, using parental DNA as a template, synthesizes new DNA strand by adding nucleotides to an RNA primer or a pre-existing DNA strand. DNA polymerase I removes RNA nucleotides of primer from 5 end and replaces them with DNA nucleotides. DNA ligase joins Okazaki fragments of lagging strand. On leading strand, joins 3 end of DNA that replaces primer to rest of leading strand DNA.